my name is Lena. I'm from the Dubois lab at UC Santa Cruz, and I'll be talking to you about my work on characterization of human astrovirus antibodies. So as you might know, antibodies are one way that bodies have to provide protection against viruses. They can recognize viral surface structures important for infection and block them from working. And there are two ways for the body to generate antibodies, either through infection with the pathogen or through vaccination. I'm focusing on the human astrovirus, which is a cause of viral gastroenteritis. There are 8 different serotypes of this virus, and we don't have a vaccine that protects against infection. So our lab is interested in developing this vaccine. On the surface of the virus is the human astrovirus capsid spike, which binds the unknown human cell surface receptor and then allows the virus to enter cells. It also elicits and binds astrovirus neutralizing antibodies, which block the virus from functioning. So because of this antibody binding capability, we believe that the spike would be a good candidate for use in a subunit vaccine that protects against astrovirus infection. In my thesis research, I have been characterizing interactions between the spike and 2 neutralizing antibodies to understand how these antibodies may target the spikes or where they bind on spike. Previously in the lab, we determined the structure of the spike from serotype 2 bound to the neutralizing antibody called PL2. And it turns out that 2 antibodies can bind the spike. So we're interested in figuring out where other antibodies will bind, which is another way of asking where are other important sites on spike that antibodies can block. So I solved the structure of the antibody called 2D9 bound to spike from serotype 8. And if you compare it to the PL2 spike 2 structure, you can see that antibody that binds serotype 8 binds it in a very different location from the antibody that binds serotype 2. That means that there are 2 separate epitopes or binding places on the spike. And that indicates that there are 2 separate sites on spike that are important for the virus to function. So in summary, we're trying to lay the groundwork for a human astrovirus spike subunit vaccine by characterizing interactions between the spike and neutralizing antibodies. I've solved the crystal structure of antibody 2D9 bound to spike 8, and in comparison with a previously solved structure, we have observed 2 separate spike epitopes. I would like to thank my lab.